Welcome to the Deep Dive. We're here to unpack complex science and really give you those essential cutting edge insights. And today, we're tackling a challenge that's right at the absolute forefront of immunotherapy. We're talking about precisely identifying T cell antigens. This is a field just exploding with innovation right now. And what we're diving into today, it's a truly groundbreaking manuscript. It was just accepted last month, May 2025. So yeah, it's really hot off the press, and honestly, it looks poised to fundamentally shift how we approach a really critical problem in biology and medicine. Think about the huge promise of personalized cancer mRNA vaccines. I know that's a topic many of you, especially if you're, third-year computational biology students, are probably following very closely. Well, the success of those vaccines, their ability to actually harness our own immune system to fight cancer, it all hinges on finding the right targets for our T cells. So our mission today is basically to give you a shortcut, a way to get quickly up to speed on this really important work. Get ready for some genuinely surprising facts, I think, and maybe a few aha moments.
And particularly for those of you really trying to bridge that gap between abstract computational models and, tangible real-world biological impact. Okay, so let's start by defining the core problem here. T-cell antigens, what are they? Well, they're essentially short peptide flags. These flags get presented on the surface of cells by molecules called major histocompatibility complex, or MHC.
Right, MHC. And in humans, we usually call them HLA, human leukocyte antigens. Same thing, different name, basically.
Exactly. And these flags, these HLA-presented peptides, they're then recognized by T cell receptors, TCRs, which are found on the surface of T cells. And that recognition event, that's the critical step. That's what triggers an immune response.
It's like a molecular handshake, a very, very specific one. It's the signal that tells the immune system, hey, look at this cell.
And we're talking about two crucial types here, and it's important for anyone building models to distinguish them. First, you've got HLA class I restricted antigens.
Yeah.
These typically come from inside the cell through what's called the cytosolic pathway. They're showing snippets of proteins made within the cell.
So that could signal things like a viral infection or maybe cancerous proteins being made, stuff the cell itself is producing.
Precisely. And these are recognized by CD8 plus T cells. You often hear them called killer T cells.
Because their job is basically to eliminate those infected or cancerous cells directly. They get the signal, they take action.
Right. Then the second type, HLA class II restricted antigens. These are generated differently through the endocytic pathway. They usually present bits of things the cell has taken up from the outside.
Like encountering bacteria or other pathogens, it's more like an external intelligence report for the immune system.
Exactly. And these are recognized by CD4 plus T cells, the helper T cells. They play a broader role, kind of coordinating the overall immune response. And the ability of these specific antigens, particularly those presented by tumor cells, to provoke really powerful immune responses.
That's the key.
That is precisely what makes them so incredibly promising for personalized cancer mRNA vaccines. It offers this path towards treatments that are truly bespoke, tailored to the individual.
Yeah, bespoke and hopefully much more effective. But what's truly fascinating here, and maybe a bit daunting, is the sheer complexity and the diversity involved. You have antigens, you have T cell receptors, and both populations are incredibly diverse. We're talking huge numbers. So it becomes this colossal needle in a haystack problem. Only a tiny, tiny fraction of all possible antigens actually trigger a meaningful, potent T cell response that we want.
Just a tiny fraction. Wow.
Oh, yeah. Identifying that fraction is a monumental task. And if we look at the traditional experimental methods for doing this, things like tetramer-associated TCR sequencing or yeast display, while they've been foundational, absolutely crucial for our understanding, they are, frankly, incredibly time-consuming.
Right. Lots of lab work, specialized equipment.
Exactly. Significant manual effort, specialized labs, highly trained people, and they're also extremely costly. Now think about the scale needed for personalized medicine. Imagine needing to identify the unique neoantigens for each individual patient's specific tumor.
Which could be thousands, maybe millions of possibilities per patient.
Potentially, yes. When you look at that scale, these traditional methods, they just aren't sustainable. They can't scale up effectively. They represent a major bottleneck in getting these therapies to patients.
Okay, so that makes perfect sense why AI would come into the picture.
Precisely. Artificial intelligence, it offers this potential to dramatically accelerate the development of personalized mRNA vaccines. How? By predicting these interactions in silico computationally. This could massively reduce the experimental burden, letting researchers focus their efforts on the most promising candidates.
But it wasn't a magic bullet right away, was it? Early AI methods still had limitations.
That's right. Even the early AI-driven computational methods, while definitely a step forward, largely relied on sequence-level features. So what does that mean for you as computational biologists? It means they typically use things like antigen sequence similarity, or maybe amino acid position-specific scoring matrices, like PSSMs, to try and estimate binding affinity. Or for the T cell receptor side, they might group TCRs by sequence similarity, using methods like GLIPH, TCR-DIS, GIANA, or deep TCR to try and figure out which antigens they might bind to.
So looking at the sequence, the string of amino acids.
Exactly. And the core problem with that approach, and this is really key for understanding the motivation behind deep antigen, is that these sequence-based methods often struggle. They struggle to efficiently capture the nonlinear relationships, the really complicated, subtle patterns involved in these molecular interactions. Fundamentally, they treated amino acids as, well, as mere tokens in a text sequence, almost like words in a sentence.
Like it's just text processing.
Pretty much. And while that's useful for finding some high-level patterns, it limited their generalizability. And ultimately, it limited their accuracy when you get down to the nitty-gritty of molecular recognition. They simply didn't see the full picture. They couldn't capture the three-dimensional dynamic dance happening at that molecular interface. They were trying to infer the dance just by reading a written script instead of actually watching the dancers move.
That's a great analogy. So if those sequence-based methods were hitting a wall, it sounds like we needed a fundamentally different approach. We needed to go deeper than just the sequence. And this is where it gets really interesting. This is where the first big aha moment from this paper, I think, really comes in. Recent research looking at actual HLA antigen TCR complex crystal structures. These are like high-resolution atomic level snapshots of these molecules interacting.
Beautiful structures, incredibly informative.
They revealed something profound. It's not just about which amino acids are there, but how they are interacting at the atomic level. Specific atomic contexts matter.
Hugely, especially non-covalent interactions, things like hydrogen bonds, salt bridges. These weak forces, when combined, really dictate the binding specificity and strength.
And they even talk about something called catch bonds. This sounds fascinating. Can you explain that a bit?
Yeah, catch bonds are really cool. Imagine like a sticky trap that actually gets stickier the harder you pull on it, at least up to a certain point before it breaks.
Yeah.
That's kind of what a catch bond does. It's a dynamic, force-dependent interaction. It actually prolongs the contact time between the antigen and the TCR, especially under the kind of mechanical stress that happens during immune cell interactions.
So the interaction lasts longer when it's a good fit under force.
Exactly. And that prolonged contact seems absolutely vital. It allows the T cell to gather enough signal to truly activate and mount an effective immune response. It's like needing a firm, sustained handshake to really get the message across, not just a quick tap.
Okay, that makes sense. But you said identifying antigens based on these atomic details has been the challenge.
Precisely. It's one thing to know these atomic interactions are crucial from looking at a few crystal structures. It's a whole other challenge to build a computational model that can predict these interactions accurately and reliably across the vast diversity of possible antigens and TCRs, especially with the limited structural data we have. That's been the bottleneck.
Right. So why have previous AI methods fallen short here, even as we gain this atomic level understanding?
Good question. The paper we're diving into today, Deep Antigen, it identifies 4 key reasons, 4 fundamental challenges that really anyone working in this computational immunology space needs to grapple with. First, there's the issue of incomplete modeling. Most computational tools developed previously, they tended to focus on just one part of the puzzle. Either they looked at antigen HLA binding or they looked at antigen TCR interactions.
But not the whole picture together.
Rarely, and not comprehensively. But you really need that full picture, the complete tripartite HLA antigen TCR complex, to truly understand immunogenicity and predict whether a T cell response will actually happen. It's the whole interaction that matters.
Okay, that makes sense. What's the second reason?
Second, and this ties back to what you were saying, is sequence insufficiency. Sequence-based methods, even sophisticated ones, are inherently insufficient. They just can't capture the refined atomic-level interactions. They miss the crucial structural nuances, the precise geometric fit, the specific hydrogen bonds or salt bridges that dictate specificity and binding affinity.
So the sequence might look similar, but the atomic interactions could be totally different.
Exactly. That's where the black box problem often comes up for biologists using these tools. The sequence similarity might be high, But if the subtle atomic interactions aren't right, you get a totally different biological outcome, binding or no binding, activation or no activation. Sequence alone doesn't tell the whole story.
Got it. Third challenge.
Third is a very practical one, data scarcity. There is a real and significant lack of high-quality, experimentally determined HLA antigen TCR complex crystal structures. These things are incredibly difficult, time-consuming, and expensive to obtain.
Right. Solving crystal structure is not trivial.
Not at all. And this scarcity severely limits the effectiveness of methods like template-based structure prediction tools. These tools rely on having similar known structures in a database to build a model from. If you don't have good templates for the vast diversity of HLA antigen TCR interactions, especially for novel neoantigens found in tumors, these tools can't accurately model them.
So not enough examples to learn from, essentially.
Pretty much. And the fourth challenge is a bit more technical, but really critical. It's about limited AI representation in existing models. Current methods often use independent encoders, separate neural network components for the antigen, the HLA molecule, and the TCR.
Okay.
This means that the representation, the sort of numerical fingerprint of a specific HLA allele, which might be capable of presenting thousands of different antigens, isn't adaptively adjusted based on the specific antigen it's presenting at that moment.
Ah, so the HLA model is kind of static, regardless of the peptide.
Exactly. It leads to a lack of flexibility, a lack of nuance in the model's understanding of that specific interaction context. It's like having a single fixed filter trying to process vastly different types of information instead of an adaptive filter that adjusts based on what it's seeing.
Okay, I see. Those are definitely significant hurdles.
They really are. And these four challenges, incomplete modeling, sequence insufficiency, data scarcity, and limited AI representation. These are precisely the complex, multifaceted problems that this new framework, Deep Antigen, was specifically designed to address head-on.
All right, so deep antigen is the proposed solution. And you mentioned it's based on deep learning, specifically graph convolutional networks, GCNs. This sounds like graph machine learning, GML, in action.
It absolutely is. GML is a really exciting area in computational biology right now, perfect for modeling molecular structures and interactions, which are inherently graph-like.
So what's the core innovation here? What's the conceptual leap that deep antigen makes, especially for, the computational biologists listening? Instead of treating amino acids as those text tokens we talked about,
Right, like just characters in a string.
deep antigen transforms the amino acid sequences of the antigen, the HLA, and the TCR into graphs, molecular graphs.
This is the fundamental shift. By representing the molecules as graphs, the model can inherently understand and process the structural and relational information that sequence-based methods completely miss out on.
It's literally looking at the molecular world through a different lens, moving from a 1D string of characters to a complex 3D web of interconnected atoms.
Exactly. And for a computational biologist, that opens up entirely new avenues for feature engineering, for pattern recognition, for understanding the underlying chemistry and physics of the interaction.
Okay, let's really dive into the mechanics of that transformation.
Yeah.
How does it actually build these graphs? Because that seems crucial to its power.
It is. So in these graphs they construct, each individual atom becomes a node in the graph.
Okay, not amino acids, but atoms.
Atoms. Carbon, nitrogen, oxygen, hydrogen atoms. Each one is a node. And the chemical bonds connecting these atoms become the edges in the graph.
So it directly represents the chemical structure.
Precisely. And this isn't just, a neat visualization trick. The fundamental insight here is that by treating atoms as these richly described data points, rather than just abstract amino acid symbols, deep antigen can see the molecular world with much higher fidelity. It can potentially uncover interactions that purely sequence-based models just couldn't even conceptualize.
It's like moving from reading the book description to actually holding the book,
Yeah.
feeling the texture, seeing the cover art.
That's a good way to put it. You get a much richer understanding of the underlying chemistry. And each atom node isn't just a point. It has a whole vector of attributes associated with it.
What kind of that?
Things like the atom type, carbon, nitrogen, etc., its formal charge, its hybridization state, sp2, sp3, whether it can act as a hydrogen bond donor or acceptor, its aromaticity, its degree, how many bonds it forms. Basically, a comprehensive chemical fingerprint for each atom.
Wow, okay, that's detailed.
And the bonds, the edges, they have attributes too, like the bond type, single, double, triple, its stereochemistry, like cis or trans, whether it's part of a conjugated system or an aromatic ring.
So really encoding the detailed chemistry.
Yes, exactly. This incredibly detailed atomic and bond-level information becomes the raw input for the graph convolutional networks. It allows the GCNs to learn these really subtle chemical and structural patterns that are just invisible if you only look at the amino acid sequence.
Okay, so you have these detailed graphs.
Mhm.
How does deep antigen use them? You mentioned GCNs and cross-attention.
Right. So once the graphs are built, Deep Antigen employs GCNs and cross-attention mechanisms to really characterize those crucial atomic level contacts between the antigen and the HLA, or the antigen and the TCR. The framework itself has three main components that work together. First, as we just discussed in detail, is the sequence-to-graph transformation, folding those amino acid strings into rich atomic graphs.
Got it.
And remember, during training, they cleverly augment this data by randomly masking a few atoms and bonds, about 5%.
Right, you mentioned that helps prevent overfitting.
It does, but it also implicitly encourages the model to identify the crucial atoms. If the model learns it can still make good predictions even when certain atoms are hidden, it suggests those atoms might be less critical. But if hiding an atom really messes up the prediction, the model learns, that atom is important. It forces it to learn the core, robust interactions. It's a smart regularization strategy for this kind of complex graph data.
Okay, makes sense. Component 2.
Component 2 is the atomic level feature extractor. This is where the graph convolutional networks really do their work. GCNs operate by iteratively updating the representation or embedding of each atom node. How? By aggregating information from its local chemical neighborhood within the graph.
So each atom is kind of looking at its neighbor.
Exactly. Imagine each atom listening to its immediate chemical environment, the atoms it's bonded to, their types, their charges, maybe even atoms a couple of bonds away, and integrating all that information into its own numerical representation.
And this repeats.
Yes, this process is repeated through multiple GCN layers. With each layer, atoms can gather increasingly global contextual information. They learn about their position and their role within the larger molecular structure, not just their immediate bonds.
Okay, so the GCNs learn features about each atom in its molecular context.
Precisely. And here's a particularly innovative detail within this feature extractor component. Within each GCN layer, they actually built a specific atomic level communication mechanism between the antigen graph and the HLA graph.
Communication between the graphs, how does that work?
It essentially allows the embedding, the representation of the HLA molecule to be adaptively adjusted based on the specific antigen it's currently interacting with or considering.
So that tackles that fourth challenge you mentioned earlier, the static HLA representation.
Exactly. It directly addresses the lack of flexibility in HLA embeddings seen in previous models. It allows the HLA's representation to be dynamically influenced by the antigen it's presenting, capturing that crucial interaction context at a very granular atomic level.
That sounds powerful. Did they test if that mechanism actually mattered?
They did. They performed ablation studies where they specifically removed this communication mechanism. And guess what? Performance dropped significantly. It really confirmed the importance of this adaptive adjustment for accurate prediction. It's a subtle, but really powerful design choice.
Okay, really clever. What's the third component?
The third component is the interacting feature map generator. So after the GCNs have done their job refining all the atom representations, this component takes over. It uses a couple of techniques: top Cadams pooling and multi-head attention.
Top Cadams pooling. What's that?
It's a way to select the most important atoms. The idea is that in complex biological interactions, often only a handful of key residues, or even specific atoms within those residues, really dictate the interaction's specificity and strength. They're like interaction hotspots.
Right. Not every atom is equally important for binding.
Exactly. So top Cat pooling helps the model focus. It selects the top K most relevant atoms based on what the GCNs have learned. This not only reduces the computational load downstream, but it cleverly allows the model to learn which atoms form these hotspots, focusing its attention where it matters most biologically.
So it's learning the critical contact points, and then multi-head attention. We hear that a lot with transformers.
Yep, similar concept. Multi-head attention allows the model to simultaneously focus on different aspects or different regions of the interaction interface between the antigen and the HLA-TCR. It helps generate a comprehensive feature map that represents the crucial atomic level contacts from multiple perspectives.
Okay, so it builds this map of important atomic contacts. What happens then?
Well, there are two outputs, depending on the specific task. Deep Antigen Sec takes these atomic level contacting features and aggregates them to predict the overall sequence level binding probability, like, does this peptide bind this HLA, yes or no? Meanwhile, deep antigen atom uses these features to directly predict the atomic level contact probabilities, like how likely is the specific atom on the antigen to be interacting closely with that specific atom on the TCR.
Wow, okay, so it can predict both the overall binding and the specific atomic details.
Exactly. And the biological intuition driving this whole architecture is powerful. Deep antigen is specifically designed to zero in on those crucial non-covalent interactions, the hydrogen bonds, the salt bridges happening between a few key atoms that really drive T-cell activation. This is the level of detail that existing sequence-based methods and even some general structure prediction tools often overlooked.
This architecture sounds incredibly well thought out, really tailored to the problem. But you mentioned data scarcity earlier, especially for the structural data needed for that atomic level prediction. How did they handle that?
Ah, yes, the data scarcity issue. This is a key challenge for any GML approach in biology dealing with structures. They tackled this head on with a really clever few-shot learning strategy. It's quite relevant for anyone listening who's facing data-limited problems. They basically did it in 2 stages.
Okay, stage 1.
Stage 1. They first pre-trained deep antigen sec. That's the part that predicts sequence-level binding, like antigen HLA binding. They used abundant sequence-level data for this data from high-throughput binding assays, which is much easier to get than crystal structures.
So training on the easier to get sequence data first.
Right, but here's the genius part. Even though it was sequence-level data, because they were using the graph representation in GCNs, the model implicitly started capturing atomic-level information from that sequence data.
How does that work?
Well, the GCN learns relationships between atoms based on the graph structure. It starts to learn patterns related to how certain amino acid combinations tend to position atoms in space, what chemical properties are nearby, even without explicit 3D coordinates. It's like learning about shapes by seeing lots of different objects, even if you don't measure them precisely. It builds an intuition for atomic arrangements.
Okay, so it gets a sort of implicit atomic understanding from the sequence pre-training. What's stage 2?
Stage 2. They froze the parameters of the GCN layers. They locked in that atomic level feature extraction capability learned during pre-training.
Okay, so the core atomic feature extractor is set.
Exactly. Then they fine-tuned deep antigen atom, the part that predicts the actual atomic level contacts, using the limited but very precise structural data available from the Protein Data Bank, PDB. These are the actual crystal structures.
Ah, so refining the atomic predictions with the real structural data.
Precisely. And this fine-tuning was guided specifically by the pairwise distances between atoms in those crystal structures. They ensured the model's predicted contact scores correlated directly with the actual physical distances. Closer atoms should get higher scores.
That makes sense.
So effectively, they bootstrapped their atomic level understanding. They started with readily available sequence data to get a broad, implicit understanding, and then refined it using the precise but sparse structural data. It's a fantastic strategy for making the absolute most of different types and quantities of biological data.
That is clever. Maximizing the value of both data types. And you also mentioned graph data augmentation. Randomly masking atoms.
Yes, during the training process, they randomly masked some atoms and their connecting chemical bonds with about a 5% probability.
And that helps with robustness and finding key atoms.
Exactly. On one hand, it acts as a regularization technique, helping to prevent overfitting. If the model can still perform well even when some information is missing, it means it has learned more robust, generalizable features, not just memorizing specific training examples. On the other hand, as we discussed, it implicitly helps the model identify crucial atoms. By observing how the removal or masking of certain atoms impacts the prediction accuracy, specifically the loss function during training, the model learns which atoms are most influential. If removing atom X significantly hurts the prediction, the model learns atom X is important.
It's like learning by subtraction, in a way.
Kind of. It's a really smart strategy for enhancing the model's robustness and its ability to generalize to new unseen molecules, which is absolutely critical if you want to apply this in the real world, like discovering new drug candidates or predicting responses to novel neoantigens in patients.
Okay, the methodology sounds incredibly sophisticated and well-designed to tackle the specific challenges. Now, for the moment of truth, let's talk results. Does this innovative GML approach actually deliver? Does it outperform existing methods?
The short answer, yes, absolutely. The paper presents really compelling evidence. Deep antigen doesn't just perform well, it consistently outcompetes the state-of-the-art methods across multiple different tasks and on diverse independent data sets. That's really what you want to see validation across different scenarios. This is the payoff for all that careful model design.
All right, let's break it down first. Antigen HLA binding prediction. This is that crucial first step, right? Getting the peptide flag presented.
Correct. And here, deep antigen showed excellent performance. For HLA class I binding, it hit an AROC, that's area under the receiver operating characteristic curve of 0.98, and an AUPR area under the precision recall curve of 0.97.
Those numbers sound really high. Close to perfect.
They are remarkably high in this context, yeah. And significantly better than well-established methods like NET-MHC-PAN or trans-PHLA. Those tools were powerful, but often relied more on sequence motifs or simpler statistical models. Deep antigen's ability to see those atomic contacts seems to give it a real edge.
Okay, high peak performance. But what about consistency? Sometimes models work well on average, but fail on specific cases.
That's a great point and something the authors addressed. What's truly impressive, especially for computational biologists looking to apply these tools, is deep antigen stability. Unlike some other methods that showed performance dips for specific HLA class I alleles or struggled with antigens of varying lengths, deep antigen maintained consistently good performance across these diverse conditions.
That's important for real-world use.
Definitely. And adding to the confidence, the model's attention maps, which show where the model is looking, actually highlighted specific anchor positions within the antigen, like the second position and the last position.
And those are known to be important for binding.
Yes, these are well supported by previous biological studies as being critical for anchoring the peptide into the HLA groove. So this isn't just about getting the right answer, it shows the model is actually learning biologically meaningful features. It's not just a black box memorizing patterns, it's picking up on real biology.
Okay, very convincing for HLA class 1. What about class 2? You said that's harder to predict.
It is, notoriously so. HLA class II molecules have a more open binding groove, allowing for more variable antigen lengths and binding modes, making prediction much trickier. But deep antigen stood out here too. It achieved the highest AUSC of 0.87 and the highest AUPR of 0.87 for HLA class II prediction among the compared methods. This represented a substantial improvement, for example, a 0.14 improvement in OROC over NET-MHC-PAN 4.2, which is a widely used tool.
A 0.14 jump in UROC is pretty significant in this field.
It really is. It demonstrated clear effectiveness in both pan-allele, working across different HLA alleles, and pan-length, working across different antigen lengths, prediction for HLA class II.
And critically, what about predicting binding for new antigens it hasn't seen before? That seems vital for neoantigen discovery.
Absolutely crucial. And deep antigen showed powerful predictive performance on unseen antigens. These were antigens deliberately held out, not present in the training data sets of any of the methods being compared. Even as the sequence similarity between these unseen antigens and the known training antigens decreased, meaning they looked less and less like anything the model had trained on deep antigen's performance remained remarkably stable.
Wow.
That speaks volumes about its generalizability. It suggests the model isn't just memorizing sequences, but has learned underlying principles of atomic interaction that allow it to predict binding even for truly novel peptides. This is paramount for personalized medicine, where you're constantly encountering unique neoantigens specific to each patient's tumor.
That stability on unseen data, that generalizability, sounds like a major advantage. Okay, so it excels at antigen HLA binding. What about the next step? Antigen TCR interactions, that recognition by the T cell itself.
Right, the second key interaction. And this is often considered even more complex to predict. One reason is what's called the long-tailed distribution of known interacting TCRs for each antigen.
Can you explain that long-tail distribution?
Sure. Imagine you plot antigens on one axis and the number of known TCRs that recognize each antigen on the other. You'll find a few well-studied antigens, maybe famous viral or cancer antigens, that have many known interacting TCRs documented. That's the head of the distribution. But then you have a vast number of other antigens, including most neoantigens, for which we know very few, maybe only one or two, or even zero interacting TCRs. That forms a long tail.
So lots of data for a few targets, very little data for most targets.
Exactly. This extreme imbalance makes it incredibly hard to train robust predictive models. The data is just so sparse for the vast majority of antigens. It's like trying to learn a language when you only have complete dictionaries for a few common words, but just single examples or fragments for most of the vocabulary.
Okay, that definitely sounds challenging. How did deep antigen handle that?
It shined here too, remarkably well, especially in some really tough testing scenarios designed to probe this exact challenge. For instance, it performed very well in zero-shot settings.
Zero shot meaning predicting for antigens it had never encountered during training at all.
Precisely. In these zero-shot tasks, deep antigen achieved an ARO-C of 0.80 and an AUPR of 0.80. That was significantly better than competing methods like PanPep, which is another tool designed for peptide MHC TCR prediction, but relies more on sequence features, and only got an ARO-C of 0.71 in the same test.
So better at predicting interactions for completely new antigens.
Yes. It also performed very well in few-shot settings, where it was given just a tiny amount of new data for a new antigen and had to quickly learn to predict its TCR interactions. And it did well in majority learning settings, too, which focus on those antigens in the head of the distribution with more known TCRs. They also did a head-to-head comparison with another method called PMTNet, using an independent test dataset provided by the PMTNet authors themselves. Deep antigen significantly outperformed PMTNet on its own test set, achieving an ARO-C of 0.95 compared to PMTNet's 0.82.
Beating another method on its home turf, essentially. That's strong validation.
Very strong. And perhaps the most impressive validation for antigen-TCR interaction was on a massive independent dataset related to SARS-CoV-2. This dataset contained over 1.1 million antigen-TCR interaction pairs. This is a huge real-world scale test, far beyond the typical benchmark datasets used in this field.
Over 1 million pairs. Wow.
Yeah. And even on this challenging dataset, where both the antigens and the TCRs could potentially be unseen by the model during its training phase, deep antigen achieved higher average ARO-C and AUPR scores than the other methods tested.
That's a massive vote of confidence.
It really is. For the computational biologists listening, this demonstrates not only the predictive power, but also the scalability and real-world applicability of this GML approach. It shows it can handle large, complex data sets, even for emerging pathogens like SARS-CoV-2, where you need rapid assessment of new antigen-TCR interactions.
Okay, so it excels at predicting both antigen-HLA binding and antigen-TCR interactions, even in challenging scenarios. But you said its most distinctive capability is predicting the atomic level contacts.
Yes. This is where deep antigen truly moves beyond what previous methods could do. Its ability to predict the specific atomic level contacts, directly pinpointing which atoms are likely interacting, is where its GML core really differentiates it. This is the granular insight, the actual molecular handshake detail that was largely missing before.
How did they measure how well it predicted these atomic contacts? It's not just a yes-no prediction anymore.
Right, it's more nuanced. They used a metric called the negative Pearson correlation coefficient, or NPCC. The idea is straightforward.
Yeah.
If the model is accurately predicting atomic contacts, then atoms that are physically closer together in the actual 3D structure should receive higher predicted interaction scores from the model.
Okay, so closer distance should mean higher score.
Exactly. A perfect MPCC would be 1, meaning predicted scores perfectly inversely correlate with distance, highest score for 0 distance. So an NPCC closer to 1 indicates the model is doing a good job capturing the distance-dependent nature of these interactions.
And how did deep antigen do on this NPCC metric?
It achieved really impressive median NPCCs, for example, around 0.57 for antigen HLA class 1 and 2 interactions, and a remarkable 0.92 for antigen TCR interactions.
0.92 is very close to 1.
Extremely close, yeah. Suggesting it's really capturing the geometry of the TCR interaction well. They also looked at it using standard classification metrics, like AUROC, by defining a contact based on a distance threshold, e.g. atoms within 5 angstroms. Here, too, the AURCs for predicting atomic contacts were exceptionally high, all above 0.81, and some, particularly for antigen TCR, were above 0.94.
So multiple metrics confirming it can pinpoint the interacting atoms accurately.
Yes, these results strongly demonstrate that deep antigen can identify those specific crucial atom pairs that exert a dominant effect on binding. And importantly, they showed that the clever few-shot pre-training strategy we discussed, learning implicit atomic info from sequence data, first significantly boosted this atomic level prediction accuracy compared to training only on the limited structural data.
So that two-stage training wasn't just for sequence prediction, it directly helped the atomic prediction too.
Absolutely. It confirms that their strategy effectively leverages the abundant sequence data to inform and enhance the precision of the atomic level predictions. That's crucial given how precious that structural data is. This atomic detail is where the model truly brings a new dimension of understanding.
And this atomic level detail isn't just, academically interesting. It has real biological implications, right? Like understanding mutations.
Precisely. This is where the rubber meets the road, biologically speaking. This atomic level detail allows deep antigen to capture the incredibly subtle effects that even single point mutations can have on T cell immune responses. Let's take a concrete example they used in the paper: the NY-ES1 antigen. This is a well-known cancer testis antigen, often targeted in immunotherapies.
Okay.
They looked at the experimental crystal structure of a specific TCR bound to this NY-ES01 antigen presented by an HLA molecule. In that structure, they observed only two specific hydrogen bonds forming between the TCR and the antigen peptide itself. One between atom V95 on the TCR and atom I6 on the antigen, and another between N97 on the TCR and T7 on the antigen.
Just two key hydrogen bonds holding them together at that spot.
In that specific interface, yes. And what's remarkable is that when they ran deep antigen on this structure, it accurately predicted the highest atomic contact probabilities for precisely those two atom pairs: TCR-V95 with antigen I6 and TCR-N97 with antigen T7.
Wow. It pinpointed the exact experimentally observed hydrogen bonds.
Exactly. That's incredibly strong evidence that the model isn't just making statistical correlations, it's learning something approximating the true molecular reality of the interaction.
Okay, that's impressive. But what about mutations?
Right, then they took it a step further. This is where the biological insight really shines. They looked at what happens computationally when you introduce mutations into that NY-ESO1 antigen specifically. They tested a W5V mutation and a Q8S mutation. They used deep antigen to predict how these mutations would change the atomic interaction probabilities with the TCR. And then, crucially, they compared these computational predictions with actual experimental data on how strongly the mutated antigens bound the TCR, how well they activated T cells, and even how effectively D cells killed target cells presenting these mutated antigens.
And did the predictions match the experiments?
The correlation was striking. Deep antigens predicted changes in interaction probabilities lined up remarkably well with the experimental binding, activation, and killing scores. For instance, it accurately showed how these specific mutations led to significant decreases in the contact probabilities between those core antigen residues, I6 and T7, and the crucial atoms in the TCR's CDR3 loop, which is the part of the TCR primarily responsible for antigen recognition.
So it didn't just predict that the mutation would have an effect, but why by showing which atomic interactions were disrupted?
Precisely. It provides invaluable biological insights. It helps explain mechanistically why certain mutations might weaken or completely abolish an immune response by pinpointing the exact atomic interactions that are broken or altered. It moves beyond a simple binding prediction to offer a rationale. It's a window into the molecular mechanism, giving us clues about why a therapy might work for one mutation but fail for another.
That level of mechanistic insight is huge. Now, this inevitably brings up the comparison question. We have these revolutionary, highly publicized tools like AlphaFold 3 and Rosetta Fold All-Atom. How does deep antigen stack up against them? They also predict structures and interactions, right?
That's a really important question for computational biology students right now. AlphaFold 3 and Rosetta Fold All-Atom are truly phenomenal tools, real breakthroughs for general biomolecular structure prediction. They are incredibly good at predicting the 3D shape of individual proteins, or even how multiple proteins might fit together in a complex. However, deep antigen seems to excel in a slightly different, more specialized niche, predicting these very nuanced interaction details, specifically the precise atomic contacts that are critical for T cell activation.
So general structure versus specific interaction details.
Kind of. Let's take that NY-ESO1 example again. AlphaFold 3 could predict the overall structure of the TCR antigen HLA complex reasonably well. It even predicted some of the hydrogen bonds correctly. But, according to the deep antigen paper, it also had issues with specific details. It predicted some false positive contacts, interactions that weren't actually observed in the experimental crystal structure.
Okay, so some inaccuracies in the fine details. What about Rosetta Fold All-Atom?
The authors of the deep antigen paper described Rosetta Fold All-Atom's performance for predicting these specific complexes as disappointing in their study. It seemed to struggle more with capturing these fine-grained interactions accurately compared to deep antigen.
Interesting. And what about predicting the effect of mutations or identifying key motifs?
This is where deep antigen specialization really seemed to pay off. When analyzing the effects of those subtle NY-ESO1 mutations or trying to identify antigen-specific TCR motifs, patterns in the TCR sequence that indicate binding to a specific antigen, deep antigen was consistently more accurate in capturing the fine-grained changes in interactions. AlphaFold 3, according to the paper, often couldn't distinguish these critical motif sites, the parts of the PCR essential for recognizing the antigen. And Rosetta Fold's predicted differences between the wild type and mutant interactions were reportedly very small, not really reflecting the significant changes seen experimentally.
So deep antigen was better at seeing the functional consequences of small changes.
That seems to be the case in these specific comparisons. It really highlights the power of having a purpose-built GML framework that's specifically optimized for analyzing these detailed molecular interactions, as opposed to a more general structure predictor. Think of it this way. AlphaFold 3 and Rosetta Fold are maybe incredible tools for figuring out what a protein complex generally looks like. Deep antigen seems to be excelling at figuring out precisely how these specific immune players interact at the atomic level, especially capturing the subtle differences that determine function, like T-cell activation. It's about having the right precision tool for a very specific but very important job in immunotherapy design.
That's a really crucial distinction for anyone working in computational biology to understand. Sometimes that specialized tool honed for a specific task can outperform even a revolutionary generalist tool for that particular application. Okay, deep antigen is pushing computational boundaries. But, as you said, the ultimate test is real-world impact. Was it tested in actual clinical settings with patients?
Yes, and this is arguably one of the most compelling parts of the paper, moving from in silico prediction to clinical relevance. They didn't just stop at benchmarking datasets, they put deep antigen to the test where it matters most, using samples from actual cancer patients. This is where computational predictions really need to prove their worth.
How did they set that up?
It was quite comprehensive. They collected tumor tissues, adjacent normal tissues as controls, and peripheral blood samples from patients diagnosed with lung, breast, or pancreatic cancer. Then, for each patient, they performed a suite of high-throughput analyses, whole exome sequencing, WES, to identify the tumor-specific mutations, the source of potential neoantigens. RNA-seq to look at gene expression levels, which helps infer if the mutated gene is actually being transcribed and potentially presented. And critically, TCR-seq to characterize the T-cell repertoires present in the patient's tumor and blood, basically cataloging the immune cells available.
So getting the full picture, the mutations, the expression, and the available T-cells.
Exactly. Connecting the dots from the genetic alterations in the tumor to the patient's immune system. Then, using deep antigen, they analyzed the WES data to predict candidate neoantigens for each individual patient, those unique peptide flags arising from the tumor mutations the deep antigen predicted would bind the patient's HLA type and potentially interact strongly with their T cells.
Okay, so they generated a list of predicted neoantigens for each patient. How did they validate them?
They moved to experimental validation using ex vivo IFN ELISpot assays.
Explain ELISpot for us again briefly.
Sure. In this context, they basically take the patient's T cells, isolated from their blood, expose them to the synthetic peptides corresponding to the predicted neoantigens, and measure how many T cells recognize and respond to each specific peptide. The response is measured by detecting interferon gamma, IFN-do, a key cytokine released by activated T cells. So if a spot lights up in the assay, it means T cells are recognizing that specific neoantigen peptide. It's a direct functional test of immunogenicity.
Got it. So did the predictions work? Did the predicted neoantigens actually activate T cells?
The results were really compelling. Across 5 cancer patients, they synthesized and tested 100 predicted neoantigens. Out of those 100 predictions, 15 successfully activated strong CD8 plus T-cell immune responses in the ELISpot assays.
15 out of 100. How does that compare to previous efforts? Is that a good hit rate?
It's actually very good in the context of neoantigen discovery, which is notoriously difficult. To put it in perspective, deep antigen achieved what they calculated as an 8% true discovery rate in this cohort. They actually identified 34 immunogenic neoantigens out of 421 total predictions across a broader analysis. This 8% rate is notably higher than the roughly 6% true positive rate reported by the Large Tesla Consortium, which was a major international effort using more established prediction and validation pipelines.
So a higher success rate in finding real T-cell targets compared to a major benchmark study.
Yes. Furthermore, deep antigen also successfully recalled 34 out of 37 known immunogenic neoantigens from benchmark datasets, showing it can find established targets effectively. And crucially, it did so with a lower false positive rate compared to other methods evaluated in the Tesla study. That means fewer wasted experiments testing peptides that don't actually work.
Lower false positives is huge for saving time and resources.
Absolutely. So this clinical validation directly demonstrates deep antigen's real potential. It's not just theoretically better, it shows promise for actually discovering effective mRNA vaccine targets or guiding other personalized immunotherapies. It's a tool with tangible clinical applicability, potentially accelerating the development of these precision treatments.
That 8% true discovery rate in real patients, beating the Tesla benchmark, that feels like a truly significant leap forward for personalized cancer treatment.
Okay, stepping back a bit now. We've seen the power of deep antigen for T-cell antigens. What does its success, particularly its sophisticated graph machine learning approach, signal for computational biology more broadly? This feels like it could have ripple effects. You're absolutely right. This is more than just a paper about T-cells. It's a landmark demonstration of what GML can achieve in molecular biology. The core success lies in its ability to capture those subtle atomic-level differences, the precise chemical interactions that distinguish an immunogenic antigen from a non-immunogenic one, or a strong binder from a weak one. This granular level of understanding is key not just for T-cells, but for many biological recognition events.
And the framework itself, the GML approach, could be adapted?
Highly adaptable. The underlying principles representing molecules as graphs with rich atomic features, using GCMs to learn interaction patterns, using attention to focus on key contexts, these are quite general. With relatively minimal modifications, you could imagine applying a similar deep antigen-like framework to predict atomic-level interactions between other types of crucial biomolecules.
Like what?
Well, think about protein interactions beyond the immune synapse, or predicting how well small molecule drugs bind to their target proteins, that's protein ligand docking, absolutely central to drug discovery. You could potentially model nucleic acid interactions like RNA protein binding or even antibody-antigen binding affinity with much greater precision than before.
So potentially a generalizable GML framework for deciphering molecular interactions across biology.
That's the exciting prospect, yes. It offers a path towards more rational drug design, more accurate simulation of biological processes at the atomic level. And beyond just prediction, deep antigen can also significantly enhance our understanding of these interaction mechanisms. By pinpointing the key atomic contacts, it helps us understand why molecules bind. This can assist in things like discovering antigen-specific TCR motifs, as we discussed, or accurately predicting the functional consequences of mutations.
And those insights have clinical value too.
Immense clinical value. Imagine being able to precisely predict how a mutation in a virus will affect antibody binding, or how changing an amino acid in a TCR will affect its ability to target a cancer cell. This is critical for applications like optimizing TCR sequences for TCR-engineered T-cell therapy, where you're essentially designing T-cells to fight disease. The more precisely we can predict and understand these interactions, the more effectively we can engineer these cellular therapies and move towards truly targeted interventions.
It sounds incredibly promising. But are there limitations? No method is perfect, right?
Absolutely, and the authors are transparent about this. Like any cutting-edge method, deep antigen has areas for improvement and inherent challenges. One practical aspect is that the initial sequence-to-graph transformation, while powerful, is computationally intensive. Building those detailed atomic graphs takes time and resources.
Could that be a bottleneck for large-scale use?
It could be, but they did show that using standard multiprocessing techniques can significantly accelerate this step, making it feasible for large-scale clinical applications, like analyzing patient WES data in a reasonable timeframe. So it's intensive, but manageable with proper compute resources. Another point is the slight information loss inherent in the top cake pooling layer we discussed. By selecting only the most important atoms, you are technically discarding some information.
Is that a major drawback?
It seems to be a reasonable trade-off. They did ablation studies comparing it to global pooling methods, which consider all atoms, and found the accuracy drop was minimal, maybe around 0.01 AROC points. But the top-cake pooling significantly reduces the computational workload downstream, so it's a pragmatic choice for balancing accuracy and efficiency. And then there's the general challenge, not specific to deep antigen, but impacting the whole field. Ongoing data availability issues, particularly for getting high-quality data on monoallelic HLA class II binding, where you know exactly which HLA allele is presenting the peptide, and especially for paired TCR alpha and beta chain data.
Right, most studies focus on the TCR beta chain because it's easier to sequence.
Often, yes. But the full TCR receptor involves both an alpha and a beta chain interacting with the antigen HLA complex. The current deep antigen study primarily focused on the beta chain due to data abundance. As more high-quality paired alpha-beta TCR sequencing data becomes available perhaps from advanced single-cell techniques or new methods like antigen-specific T-cell sequencing, incorporating that full pairing information is expected to improve deep antigens performance even further.
So performance could get even better as the input data improves.
Exactly. Which leads to an interesting thought, especially for the students listening. How will this continuous accumulation of better, more comprehensive data, like neoantigen-specific paired TCR sequences, fuel an iterative cycle? You'll have better data leading to better algorithm optimization, like refining deep antigen, which leads to better predictions, guiding more targeted experiments, which generate more high-quality data.
A virtuous cycle.
Precisely. How will this cycle accelerate the discovery of truly immunogenic neoantigens and the overall development of breakthrough immunotherapies? It feels like we're on the cusp of a rapid acceleration phase, driven by the synergy between advanced computational methods like GML and improving experimental technologies.
That's a really exciting prospect. So wrapping up, what we've really seen today is deep antigen pushing the boundaries in T-cell antigen identification. It's leveraging these powerful graph convolutional networks to move beyond just sequence analysis, getting right down to the atomic level of interaction. This allows for unprecedented precision in understanding these absolutely critical immune interactions. And crucially, this isn't just some theoretical advance stuck in a computer. It's already showing remarkable proof of principle in clinical applications, holding real promise to revolutionize personalized neoantigen-targeted immunotherapies, cancer mRNA vaccines, the whole field, really. So for you, the computational biology student listening, here's maybe a final thought to mull over as you continue your studies. How will this convergence, this meeting point of increasingly sophisticated graph machine learning methods like deep antigen and the accelerating generation of high-quality biological structural and interaction data fundamentally reshape our very understanding of molecular recognition? How will it accelerate the entire pipeline of therapeutic design in the coming years? What completely new insights, what new therapeutic strategies might emerge when we can truly precisely model life down at the atomic scale, finally seeing all the intricate details of those molecular handshakes. That's all for this deep dive. Until next time, keep exploring, keep learning, and definitely keep asking those fascinating questions.